In this video, we're going to talk about the structure and function of carbohydrates. The material from this slideshow is mostly sourced from Cognitive Higher Level Biology from our online textbook. Before we talk about carbohydrates, we really need to talk about carbon and its properties. So carbon has a binding capacity of 4. It can covalently bond with 4 other atoms because it has 4 valence electrons and is looking to fill that to fulfill the octet rule. It forms covalent bonds with other nonmetals, with itself, or hydrogen, oxygen, nitrogen, carbon are the most common. Phosphorus, sulfur can be other ones as well. It can form single, double, and triple bonds with other atoms, so that gives some versatility to the molecules it can make as well. And then finally, it can form long chains of carbon atoms, which can be found branching, or it can also form rings as well. And all of these will be seen in the biological molecules that we will be looking at this unit. So you may be familiar with this term, but we'll review it again. Polymerization is when molecules can join together to form long and branching chains. Not necessarily branching, but can form long chains, sometimes branching. One example would be glycogen, which is the molecule to the right. It's a polysaccharide that's found in animals. So let's discuss what a functional group is. A functional group is a group of atoms that is attached to a carbon chain, and it'll give that molecule certain properties, whether it'll make it hydrophilic or hydrophobic. It may make it soluble in water or insoluble in water. It can also help us identify different types of organic molecules. When you take your organic chemistry unit, you'll learn more about all the different types of organic molecules. But 2 of the main functional groups, the ones that we'll see in carbohydrates, are the OH, hydroxyl, and the double bonded O, which is the carbonyl or carbonyl group. Macromolecules are molecules that are made of smaller subunits. So carbohydrates, lipids, nucleic acids, and proteins are all examples of macromolecules. And smaller subunits are joined together through what is known as condensation reactions, which we discussed in our last lesson on water. One subunit will lose hydroxide, and one will lose a hydrogen, and that will form a new molecule that's been condensed together, hence the word condensation, plus a water molecule. So there'll be some condensation formed when these 2 molecules condense or build together. However, to be able to be used by the body, macromolecules need to be broken back down into their subunits and then acted on by enzymes. These reactions are done by hydrolysis, and again, enzymes are responsible for this. And hydrolysis adds water across the bond to form 2 molecules. And you can see that in the reaction below. So AB plus water makes AH and BOH. So the water splits the bond and then reforms with the hydroxide and the hydrogen. So now let's get into carbohydrates. Carbohydrates are made of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So 1 carbon to 2 hydrogens to 1 oxygen ratio. They're made by plants by photosynthesis. Sources would be fruits and vegetables for the most part. Some uses would be short-term and long-term energy, depending on the type of carbohydrate. And also some structure too. So in plants, cellulose is a structural carbohydrate. Its job is to form the cell wall and to keep rigidity to the cell. So a couple of fun facts about carbohydrates. The names of carbohydrates often end in OSE, so glucose, fructose, sucrose. So if you see OS in a food label, you know that that's a carbohydrate. And they're found in the form of monosaccharides, disaccharides, and polysaccharides. And the word saccharide is Greek for sugar. So monosaccharides, they're the simplest sugars. They're made of 1 carbohydrate molecule. The most common ones, and the ones that you'll probably hear have referred to over and over again in our unit would be glucose, which is the primary energy source of all cells. Fructose, which is a fruit sugar, so it's found in fruit. It's much sweeter than glucose, which makes fruit sweeter. Galactose is found in milk. Deoxyribose is found in DNA. So the structure of monosaccharides. They form a single carbon chain, and on every carbon there's an OH group attached to all the carbons except for one. And the arrangement of the hydroxide groups or the OH groups help us identify the sugar, and we'll see that a little bit later when we look at some examples. One of the carbons has a carbonyl or carbonyl group attached to it, and that monosaccharide forms a ring structure. 5 carbons would be a pentose sugar, 6 carbons is a hexose. You could even have a triose sugar if you wanted. So what are isomers? Isomers are molecules that have the same molecular formula but different arrangements of the atoms. So if you counted up all the atoms on these 3 molecules found here, glucose, galactose, and fructose, they would have the same number of carbons, hydrogens, and oxygens. Now, glucose and galactose look very similar. And if you remember from the last slide, we talked about how the different monosaccharides differ on where the OHs are found. The only thing that you see different between glucose and galactose is on this carbon right here. The OH points up, and on this one, the OH points down. Now, there's an interesting thing about carbohydrates. When we name these molecules, or we refer to what carbon the hydroxides are bonded to, we always start counting the carbons. We number the carbons. We start clockwise just to the right of this, the oxygen that's in the ring. So this would be carbon 1, this would be carbon 2, this would be carbon 3, this is carbon 4, 5, and then 6 points up at the top. Over here in galactose, same thing. So this is carbon 1, 2, 3, 4, 5, 6. So one thing that we need to know about are the isomers of glucose. So in alpha glucose, the hydroxide on carbon number 1, remember how we talked about the numbering, points down. And in beta glucose, the OH points up. This impacts the polymers that can form between these 2 different types of glucose. So starch and glycogen are polymers of alpha glucose, and cellulose is a polymer of beta glucose. Let's talk about the solubility of glucose. Glucose is very soluble in water because of all of its hydroxyl groups or OH groups, which are polar. In solution, the OH on carbon 1 is in flux. It doesn't form into alpha or beta glucose until the molecule forms a polymer, so it could be either or. And because glucose is polar, it's very easily transported in the plasma of blood, which is really important because we need to deliver this essential nutrient to every cell of the body for energy usage. Next unit, when we learn about cellular respiration and photosynthesis, we'll learn about glucose's important role in providing energy. Glucose is also a very stable molecule. Because it's found in a cyclic molecule with the hydroxides on each carbon in the ring, it's very stable, and it helps to form stable polymers as well. So it's not going to be very reactive. It's going to remain until it's needed for energy usage or for structural support, depending on the type of polymer it's forming. Again, I briefly touched on the important use of glucose in cellular respiration. What happens in the series of reactions is that glucose gets oxidized to carbon dioxide, and those oxidation reactions release energy in the form of ATP, or adenosine triphosphate, and that's the energy molecule that drives all the reactions in our body. We'll learn more about it a little bit later, but we sometimes call ATP the currency of the cell. We can't get anything in real life without money. You can't get anything or do anything in the cell without ATP. And we'll explore those reactions in a later unit, as I said. So disaccharides are two joined monosaccharides, and they're made through condensation reactions. You'll hear that phrase come up over and over again. And the bonds between the monosaccharides are called glycosidic linkages. Here's one example. So this is the example of the reaction between two alpha glucose to make maltose. So they come in close proximity to one another. Hydrogen comes off of one of the hydroxyl groups and an OH comes off the other, and then you end up with this molecule, maltose. Maltose is when you join two glucose together, and you find maltose in grains, and it's also used to make beer. Sucrose is a glucose and fructose. That's table sugar. So the sources of table sugar would be, for commercial use, would be sugarcane, beets, maple trees, and so on. And then lactose is glucose plus galactose, and that's the sugar that we find in milk. And that happens to be the sugar that some people are not able to digest anymore, and that is what leads to lactose intolerance. Polysaccharides are complex carbohydrates, and they can be anywhere from several hundred to several thousand monosaccharides long. And they can play either a structural role or energy storage role. The first one we'll talk about is starch. It's a long-term energy storage molecule that's found in plants. So plants store up glucose in the form of starch, and then when energy is needed, glucose molecules will be broken off from the longer starch molecule and be used for energy. And starch is a long branching chain of glucose molecules, and it's generally stored in the roots and the stems of plants. And there's two different forms of starch, amylose and amylopectin. So here's a look at the two different ones. Amylopectin is quite branching, like so, and it actually is harder to break down and it's less soluble. So pectin, you may have heard of, is something that we use to thicken jam when you're making jams. You may add pectin to make jam, and that's because of its lack of solubility. And then amylose is a little more soluble. It still isn't very soluble, but it's more so than amylopectin. So in amylose, glucose is connected through alpha-1,4 glycosidic linkages or bonds. It doesn't branch, but it does form a coiled structure. It is insoluble, and it's approximately 300 to 3,000 glucose molecules long. Amylopectin is branched, and the glucose gets connected through alpha-1,4 linkages and then alpha-1,6 glycosidic bonds where the branches are forming. And this forms a 3D structure that can store energy much more effectively, and it makes up about 80 to 85% of all the starch molecules. And it, like I said, is insoluble as well. Glycogen is an energy storage molecule in animals, and it forms alpha-1,4 glycosidic bonds in a coil form with alpha-1,6 branches about every 8 to 12 glucose molecules long. And it's even more branched than starch, than amylopectin. And the excess glucose in our blood gets stored in our liver and muscles as glycogen. And then when our glucose levels drop in our blood, it gets broken down into glucose molecules for energy. So that's why we don't have to eat continually. We can actually store up some of our glucose stores in the form of glycogen and then release it as we start to need more glucose in our body. Cellulose is a structural polysaccharide for plants, and it helps make up the cell wall. And it's a straight chain polymer beta glucose. Remember, all the other starch and glycogen were alpha glucose. Cellulose is a polymer beta glucose. Several of the chains are held together by hydrogen bonds, which forms microfibrils. And then the microfibrils intertwine to form fibers. And this type of polysaccharide is not digestible, so this is what we call fiber and in our diet, and it provides strength and rigidity to the cell wall, and it's very important in the structure of plants. Now, glycoproteins are proteins that have at least one carbohydrate bonded to them, and they're very important in cell signaling in a variety of different ways. First of all, they can be markers for cell-to-cell recognition. Secondly, they can be receptors for hormones and other molecules. Thirdly, they can be ligands for molecules to bind to. And fourthly, they can provide the support and structure by forming the extracellular matrix. One type of glycoprotein you may have learned about already, but didn't know it was a glycoprotein, are ABO blood groups. So blood cells have glycoproteins, can have glycoproteins bonded to their cell surface, which leads to the ABO blood groups. Type O blood has no glycoprotein on the outside. Type A blood has blood cells with the type A glycoprotein. Type B blood has blood cells with the type B glycoprotein, and type AB blood has both A and B glycoproteins. So you can't receive blood from a donor with a glycoprotein that you don't have. So blood type O is considered the universal donor because any type of person can receive blood type O, but blood type O can only receive blood type O. So while it's a universal donor, it's not a universal acceptor. The universal recipient is somebody with blood group AB because they could get type O, type A, type B, or type AB blood.